Negative staining is a type of simple staining. In this case, the background is colored and the cell remains colorless. In negative staining, heat is not applied to affix the bacterial samples on the slide, so bacteria are less distorted. This is one method of negative staining. Your instructor may ask you to follow a different procedure, but the end result is the same. You will need 2 slides for this experiment. Place a small drop or loopful of nigrosin at the end of each slide. For cultures on solid media, add a loopful of distilled water and emulsify a small amount of the culture in the nigrosin water drop. Flame the loop, get a loopful of water, and add it to the drop of nigrosin. Flame your loop, remove the cap from the tube containing the solid culture. Pass the tube through the flame, obtain a loopful of bacteria, and mix it with the nigrosin water drop on the slide. Using the end edge of another slide, spread out the drop to produce a smear varying from opaque black to gray. The angle of the spreading slide will determine the thickness of the smear. Let the smear air dry. Do not fix it. For broth cultures, mix a loopful of the culture into the drop of nigrosin. Using the second slide, flame your loop, remove the top from the tube containing the broth culture, pass the tube through the flame, and obtain a loopful of broth. Mix the loopful of broth with the drop of nigrosin in the second slide. Using the end edge of another slide, spread out the drop to produce a smear varying from opaque black to gray. The angle of the spreading slide will determine the thickness of the smear. Let the smear air dry. Do not fix it. Examine the slide under the microscope. On areas of the slide where the stain is too thick, the smear will appear cracked and is not viewable. Likewise, areas that have too little stain will not provide enough contrast with the bacteria. Find the area that shows colorless cells against the black background. Nigrosin is a negatively charged dye, and so is the bacterial surface. This causes the dye to be repelled. The background stains a dark color with a clear cell and capsule. When you have finished viewing your results, gently blot the oil from the slide and store it in a slide box.